My name is Dave Penrose. I'm the current chair of the Society, the Council of Scientific Society Presidents. And I am reminded of the fact that in the late 1870s, early 1800s, that scientists demanded and received auditoriums full of people, wild rock star-like people. And I'm so encouraged to see this audience, such a large and enthusiastic audience for this presentation. So I must say, Dr. Doudna, that you must be a rock star. I have a prepared statement, and I'm just got a new pair of glasses, so this might be, we'll see how this goes. The Council of Scientific Society Presidents, or CSSP, is a unique organization representing the breadth of science, engineering, research disciplines throughout its member societies and federations. CSSP member societies are represented by their presidents, presidents-elect, and recent past presidents of leading scientific societies and federations whose combined membership totals over 1 million scientists worldwide. Founded in 1973, CSSP has served as a Center for National Science Leadership Development, a strong voice in support of science, and the premier forum for national science policy development, and open, substantive exchanges on current issues encompassing the full spectrum of science, engineering, and mathematics. CSSP is committed to advancing leadership in science and technology and to establishing policies and programs that will ensure a bright future for the 21st century science. And now it is my honor to introduce tonight's special speaker, Dr. Jennifer Doudna, who is the Fred Kavli Science at the Frontiers Lecturer presented by the Council of Scientific Society Presidents through the generosity of the Kavli Foundation and in collaboration with the Carnegie Institution for Science. Dr. Doudna is the Li Ka-shing Chancellor's Chair in Biomedical and Health Sciences, and she is a professor of chemistry and molecular and cell biology at the University of California at Berkeley. She has been an investigator with the Howard Hughes Medical Institute since 1997, and is a member of the Physical Biosciences Division at Lawrence Berkeley National Lab. Dr. Doudna received her BA in chemistry from Pomona College in 1985, her PhD in biochemistry from Harvard University with Jack W. Sostak. She did her postdoctoral work with Thomas Cech at the University of Colorado in Boulder. She joined the faculty of Yale University in 1994. She was promoted to the position of Henry Ford II Professor of Molecular Biophysics and Biochemistry at Yale in 2000 and joined the faculty of the University of California at Berkeley in 2002. Dr. Doudna has received many honors and awards. She is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the National Academy of Medicine, and the National Academy of Inventors. She is a recipient of such prestigious awards as the NSF Waterman Award, the Lurie Prize from the Foundation for the National Institutes of Health, and with Charpentier, the Paul Janssen Award for Biomedical Research, the Breakthrough Prize in Life Sciences, the Princess of Asturias Award, and the Gruber Prize in Genetics. She's also received the Massey Prize and the L'Oreal UNESCO International Prize for Women in Science. Her Kavli lecture is titled CRISPR Biology and the New Era of Genome Engineering. Her research seeks to understand how RNA molecules control the expression of genetic information. Her research led to insights about CRISPR-Cas9-mediated bacterial immunity that enabled her lab and that of her collaborator, Emmanuel Charpentier, to design this system for efficient genome engineering in animals, plants, creating a transformative technology that is revolutionizing the field of genetics molecular biology, and medicine. Science magazine named this genome editing technology Science Breakthrough of 2015. Dr. Doudna was also named one of Time magazine's 100 most influential people in the world, together with Charpentier. I know we are going to enjoy this talk tonight. After Dr. Doudna speaks, after Doudna speaks, we will have a question. It was bound to happen, the very last paragraph. Sorry. I know we are going to enjoy this talk. We will have a question and answer session moderated by Frank Sesno, an award-winning journalist who is director of the School of Media and Public Affairs at George Washington University, following your presentation. So with no further ado, Dr. Doudna.
Well, good evening, everyone. It's really a pleasure to be here with you, and I first want to thank the CSSP, the Kavli, and the Carnegie for the opportunity to come and talk to you about our science and about my thoughts about sort of where this technology is going in the future. And the story I want to tell you tonight, as you'll see, is really a story, I would say, of small science, and science that began in a very different place than it ended up. And that's partly what makes it near to my heart, is that I've always been a scientist interested in the fundamentals of biology. And through the course of our research and curiosity-driven questions about how bacteria fight viral infection, we were able to uncover a pathway that can be now harnessed as an exciting technology for changing the DNA inside of cells. So I want to begin by, slides here. So I want to begin by just sort of describing what we think about as biologists in terms of understanding cells and organisms, and then share with you my own journey on the path of understanding bacterial adaptive immunity and how that led to the CRISPR technology for genome engineering. And so I wanted to start it off by just pointing out, for those of you that don't think about this every day, that DNA is the code of life. It's the molecule inside of cells that encodes all of the information that cells need to grow and divide and become a tissue or become a whole organism. And so ever since people have been trying to understand the molecular basis for life, we've really always been sort of drawn back to DNA at some level. And when we think about DNA, we can't help but be awed by the beautiful structure of this molecule. And again, to remind you that this is a molecule that consists of two strands of chemical letters that wind around each other, and they form a double helix, as was shown in the 1950s. And what makes this structure particularly both beautiful and informative about biology is that the chemical letters in the DNA pair with each other in such a way that we can understand how one strand of DNA in the double helix can be copied into the other strand, because one letter always pairs with the other, A's with T's, G's with C's. And ever since the discovery of the structure of DNA and the understanding of its importance in biology, scientists have had the idea that wouldn't it be amazing if one could actually manipulate or change the DNA in cells? Because we know that changes in DNA lead to changes in cells and changes in whole organisms, and in humans, of course, can lead to genetic diseases and to differences between all of us. So the thing I want to pose to you, the question I want to pose to you, and what I want to talk to you about tonight, is imagining that we had a technology for changing the DNA inside of cells, much like we might change the words in a text document. So you know that in your word processor, if you see a typo in the document, you can easily fix it by replacing a letter or a word or moving words around. What if you could do the same thing with the letter code of DNA inside of cells? And I would say that science is really, biology has really been working in that direction for decades, effectively, by understanding how cells manipulate DNA. Of course, the work of Barbara McClintock here at the Carnegie was one of the first pieces of her work showed that DNA is very much more dynamic than people appreciated at the time. And what I'll show you tonight is that we now have a technology that really brings us to this point where we can control DNA at the level of a single letter in the entire DNA sequence of a human cell with 3 billion base pairs of DNA. And where did this technology come from? It really came about not through a focused effort to discover how to do this, but actually from something that sounds completely unrelated and sort of obscure, which was the question, how do bacteria fight off viral infection in their environment? And so I want to point out that bacteria, like most other organisms that we know about, including humans, face viruses in their environment all the time. And when a virus lands on a cell, in this example, I'm showing you an example of a virus landing on the surface of a bacterial cell, the way it infects the cell is actually to inject its genetic material, in this case its DNA, into the cell. And then the DNA, of course, contains the program required to build more viruses. So it takes over the cell's machinery and begins to build more viruses, and that eventually leads to destruction of the cell. So bacteria, like other organisms that have to face this threat in their environment, have evolved a number of pathways to fight off viral infection. And a pathway, you might think that bacteria have been studied for a long time and that we already know all about this, but it turns out that about 10 years ago, a new pathway for bacterial defense against viruses came to the attention of just a few people who were looking into this in the biological world, and that was a pathway called CRISPR. And so to explain this, I want to just point out, so this is a cartoon that illustrates, so the black squiggly line is meant to represent the circular DNA molecule or genome inside of a bacterial cell. And a colleague of mine at the University of California, Berkeley, named Jillian Banfield, was working away on understanding bacteria in interesting environments that were not in the laboratory. So her research group goes to places where bacteria are growing at pH 1 and at very cold temperatures or very high pressures, things like that. Many of these organisms have never been identified. It's not possible to culture them in the laboratory. So the way she studies them is to collect samples of those organisms and then sequence all of the DNA that's present in the sample. And by piecing together the DNA sequences found in those samples, she could actually reassemble these kinds of circular chromosomes and figure out what the DNA code of those organisms is, and also what kinds of viruses they're interacting with. And her research led to an interesting observation that a few other people doing similar research were making at around the same time. And that was that in many bacteria, there's an interesting pattern of DNA sequences inside the chromosome of the cell. And what I'm showing you in this slide is the pattern represented by black diamonds, which are DNA letters that are sequences of letters that are repeated over and over again. And in between the repeats are sequences that are unique in the chromosome, and in fact sort of unique in the bacterial world. And so a few labs had noticed these types of sequences. Nobody knew what they were. They had been called CRISPRs, which is an acronym that stands for clusters of regularly interspaced short palindromic repeats, a big mouthful, but represents this interesting pattern of DNA sequences. And in 2005, Jill Banfield called me in my office one day to tell me that not only was she identifying a lot of these CRISPR sequences in bacterial DNA samples, but also that the first clue to what they were doing had been found, which was namely, in work published that year, the observation that the unique sequences represented by the colored boxes come from viruses in the environment of these organisms. So it really was the first hint that what these unusual sequences were is actually effectively a genetic vaccination card for cells. The way cells can keep a record of DNA from viruses that have infected them over time and pass that information along to their progeny. And so the question was, why do they do that? And one idea that came up in discussions among a few scientists at the time was that maybe these actually represent some kind of bacterial immune system, a way that bacteria can acquire DNA from viruses and then somehow use that information to protect cells in the future. And what made it look suspiciously like some kind of a conserved system in bacteria was that next door to these repetitive loci called CRISPRs, there were very typically Cas or CRISPR-associated genes that encode proteins that were found neighboring these repetitive sites. So it really looked like some kind of a pathway, perhaps, that cells had maintained for a purpose. So why did Jill call me about this? And to explain that, I have to make a little segue to tell you about research that I've been interested in for my whole career, which is namely the function of molecules called RNA inside of cells. And I'll just preface this by saying that Jill Banfield at the time suspected that the way these viral sequences might be employed in cells was at the level of RNA, to help recognize viral molecules that could get into the cell and try to infect cells, and somehow use that information to protect the cells. So to explain what I mean by that, I want to just show you a slide that illustrates what we call the central dogma of molecular biology. And this is greatly simplified from what we now understand actually happens inside of cells, but for students that have taken a biology class in high school, this is often what they learn, is that on the left-hand side, we have DNA molecules that store genetic information inside cells. They're responsible for replicating that information accurately and passing it along to progeny cells. And the way that the DNA information is actually employed in the cell is by the cell copying it into other molecules. So first of all, copying it into molecules called RNA, which have the same type of sequence of letters that can form interactions with DNA sequences of letters, very much sort of to form helical structures like you see for DNA. And in many cases, these RNA molecules encode proteins, so they can be translated into proteins that become the functional molecules of the cell. But I've always been interested in sort of this process shown right here, which is when the RNA molecules that are copied from DNA are actually functional in their own right. They aren't simply serving as the code for proteins, but they actually form interesting structures and they do interesting things on their own. So knowing this about me, Jill said, maybe these CRISPR sequences are being converted into RNA molecules that are being employed in bacteria to recognize viruses. So I got intrigued by that possibility. And so in a very small scale project, just a couple of people in my lab that started working on this, we started to investigate this hypothesis about a bacterial adaptive immune system and how it might operate. And what emerged over the next several years in research from just sort of a handful of labs around the world at the time is that bacteria that have a CRISPR sequence indeed have the ability to adapt to viruses that infect them. And this slide shows 3 steps in the adaptive immune system. First of all, detection of foreign DNA that gets injected into the cell, say from a virus, incorporation of little bits of the viral DNA into the CRISPR sequence, and then a copy is made of that that sort of recorded sequence of viral DNA in the form of RNA that can assemble with proteins to form guided, sort of RNA-guided complexes that search through the cell. They're like sentinels that go looking for DNA sequences that have a match to the letter sequence in the RNA, which remember comes originally from the virus. And when a match is found, That allows this interference complex to grab on to the viral DNA and allow the associated proteins to make a cut in the DNA. So it's a fascinating way that bacteria can actually acquire immunity to viruses by storing their DNA in a chromosome. And so that led to a series of experiments that we worked on in my lab to understand how this actually works. And eventually to a chance meeting that I had with Emmanuel Charpentier, who is a medical microbiologist, who was studying a particular type of organism that had a very interesting CRISPR system in its chromosome, in which there was a single Cas gene called Cas9 that had been identified genetically at that time to be critical for the adaptive immune system to function in that organism. And so when she and I met at a conference in 2011, we decided to put our complementary types of research expertise together to collaborate to figure out the function of Cas9. And so in a really fun collaboration that was carried out really across many thousands of miles with her student Krzysztof Czajlinski in Vienna at the time and Martin Janek, a postdoc in my lab in Berkeley, these two scientists teamed up to test the function of the Cas9 protein. And what they figured out, and is diagrammed here on the slide, is that the Cas9 protein, which is shown in the blue, sort of that blue blocky structure, is a protein whose job is to grab onto DNA at places in the DNA sequence that match a 20 nucleotide or 20 letter sequence in an RNA molecule, which is the, I don't know if I can point here, I guess I can use this. Here we go. Yeah, right here. So this molecule right here. So here's the guide sequence in the RNA, and this is an RNA molecule that would originally come from the CRISPR locus. So this would be a sequence that would be originally derived from the virus in principle. And when this protein finds a matching sequence to this RNA, it unwinds the DNA double helix to allow two molecular blades in the protein to make a cut in each strand of the DNA. So it breaks the DNA, and in bacteria, that allows the DNA to eventually be destroyed. Now, we figured out a couple of things that were very interesting about the way this chemical reaction with DNA operates. First of all, we found out that it doesn't work with just this RNA molecule right here. We had to have a second RNA called tracer, which is the red molecule, that is important for forming a structure in the RNA. Again, this is a base pairing interaction, very much like the double helical DNA structure that I showed you, except it's an RNA. And that structure binds to the Cas9 protein and allows this whole targeting complex to assemble. So you have to have that second RNA molecule. And so Martin Janek was a very good biochemist. He was trimming away at these natural RNA molecules to figure out what was minimally required for this reaction to work in the test tube. And that led to the realization that we could simplify the system compared to what nature has done. And the idea was to link together the part of this RNA molecule that provides the DNA recognition information on this end with the part of the RNA that's important for assembly with the Cas9 protein over here to create a single guide RNA molecule that could bind and form this sentinel complex with the Cas9 protein and have the targeting information contained right here. And why was that interesting? Well, once Martin had done this experiment, had this idea, and did this experiment in the lab, we found out that we could very easily program this protein, which is sort of like a molecular scalpel, to find and cut right here. And because this is now a single RNA, it was a two-component system, a single protein and a single RNA. The RNA acts like a programmer for the protein, can be easily changed to direct the protein to a different piece of DNA to make a cut. And for us, that was really the moment when this project went from a fundamental research project, a curiosity-driven discovery, to recognizing that we could actually harness this as a really exciting technology. And to make that connection, I want to show you a couple more things. So first of all, how does this DNA recognition actually work? And I want to show you here a 3D printed model of the Cas9 protein. So of course this complex, how small is it? It's really small, right? We can't really see it we certainly can't see it with a light microscope. We can see maybe little specks using an electron microscope. But to really understand the molecular structure of this complex, we have to use a technique called X-ray crystallography. And this model is built using actual atomic coordinates that come from an X-ray crystallographic structure. And so what you're looking at here is the protein in white, and its guiding RNA is the orange molecule. And that complex searches through cells looking for a DNA sequence that matches the sequence on the end of the guide RNA. When that occurs, it grabs onto the DNA, so you can see this beautiful double helical structure here running through the protein. And at the place where the sequence in the DNA matches the RNA, an RNA-DNA helix forms inside the protein. That's how recognition actually occurs. And that displaces the other strand of the DNA and positions two molecular blades in the protein to make a precise double-stranded break in the DNA. So it's a cleaver that comes in and cuts the DNA when this kind of chemical interaction actually occurs. So that's great, but how does that relate to what we talked about earlier, genome engineering? And to explain that, I need to show you that this sort of connects to a long and very interesting line of research by many, many labs that have been studying how plant and animal cells, and our cells, deal with double-stranded DNA breaks, which occur all the time naturally during cell division, but also can occur due to DNA damage. And so cells have evolved very nice, sophisticated machinery to repair double-stranded breaks in DNA. And in this example, so here, if this is a segment of DNA inside of the cell, if it gets broken, cells can repair the break by repair pathways that include a mechanism that chemically pastes the ends back together, sometimes with a small disruption in the DNA sequence at the site of repair, or in some cases integrates a new segment of DNA at the site of the break to actually incorporate new DNA sequence into the DNA at exactly the place where it was originally broken. So many scientists had appreciated that if you wanted to alter the DNA sequence inside of cells, the way to do it would be to figure out how to introduce double-stranded breaks at places in the DNA where you might like to effect a change. And several very interesting strategies have been developed, particularly recently, for doing this that involve engineered proteins. Proteins that, you don't have to worry about these acronyms here, but engineered proteins that can be designed in the laboratory to be able to bind to particular DNA sequences. And I can't help but saying, I know Carl Pino is here tonight, and zinc finger nucleases right here were actually based on work that his lab originally did to understand exactly how these proteins interact with particular DNA sequences or DNA letters. These can be very effective technologies, but they had not been widely adopted for genome editing. The reason was they just require a high enough level of sophistication about protein engineering, and time and money to develop proteins for every experiment, that many labs, including my own, had looked at those technologies, thought they looked really exciting, but didn't feel like we actually had the expertise and the money and the time to actually deploy them. And so with Cas9, the idea was here we have a single protein that can be reprogrammed for different DNA sequence recognition and cleavage by simply changing the short sequence of guide RNA that it binds to, something that any student, even of molecular biology, can do. And these days, they type these sequences into their iPhone and they can mail it off and get the DNA or RNA molecule the next day. So it's a trivial thing to do. So that was the idea. And so what happened next was truly amazing. So we proposed this in a publication in 2012, and before the end of 2012, there were already 6 manuscripts submitted to scientific journals that had had employed this method for engineering a variety of different kinds of cells, including human cells, human stem cells, mouse cells, plant cells, and very quickly thereafter, many labs around the world began to adopt this technology for genome engineering in their system of interest. And so why did this technology take off so quickly? And I really like to point out three sort of points that kind of explain this, I think. One is what I like to call software versus hardware. So where earlier technologies were hardwired, requiring new protein engineering for each experiment, here we had a system that's more analogous to software. A single protein reprogrammable by using these short sequences of RNA. Lots of different kinds of applications in various systems, and as we've discovered over the last 2 years investigating how this actually works, a rapid and accurate DNA targeting mechanism that really lends itself to harnessing as a technology. So let me show you a short video that illustrates how we imagine that this system operates inside of animal and plant cells, and also points out some of the questions that we still have about how this bacterial system is functioning inside a cell that has a nucleus, like a human cell, for example. And so this is showing, this is a video made by Janet Owasa at the University of Utah for us. We're zooming into a cell, and of course, in a plant or animal cell, the DNA is inside the nucleus, so we're going inside the nucleus, and the DNA is highly packaged in chromatin, these structures called nucleosomes. And somehow, this bacterial enzyme Cas9, with its guide RNA, has to search through all of the DNA of the cell to find a single sequence that matches the sequence of the guide RNA. When that occurs, it binds to the DNA, unwinds it, and forms that structure that I showed you, cuts the DNA, and then hands off the cleaved ends of the DNA molecule to repair machinery, repair enzymes in the cell that can fix the break, and in that example, actually insert a new piece of DNA at the site where the break originally occurred. And remarkably, this system operates in effectively any kind of cell where people have tried to employ it. So it's a very democratizing technique, it's very inexpensive to use, and allows scientists to now make very precise changes to the DNA of cells for research and other kinds of applications. And I just want to now point out sort of what people are doing with this. So it's been used in a wide variety of different systems, plants and animals that are important for agricultural animals we like to have as pets, and for engineering organisms like fungi that are used for things like green chemical production or sustainable biofuel production. Just about 2 weeks ago, I opened up Nature magazine, and there was this article about a company in the Midwest that has made a CRISPR mushroom. This is a mushroom that doesn't brown when you cut it, because they were able to knock out or disrupt a single gene that's important for that browning process. And why this was in science was actually not that they had made this mushroom, but actually that the USDA had decided that it would not be regulated because it doesn't contain any foreign DNA. So in the US, this would not be considered a genetically modified organism. Shortly after that, I got an email from a prominent company that is in the agricultural sector informing me that they had 25 plants of this nature in the pipeline, engineered with CRISPR, with products that will be coming out very soon. And they were, of course, thrilled that the USDA is not regulating this application. So what about in humans, and what about for biomedicine? So, we understand that now that we can use the CRISPR system for engineering cells like stem cells that might be useful for treating disease in the future, animals that are important for modeling human disease, and maybe in the not too distant future for treating perhaps actual patients. And there were 3 publications that appeared in Science magazine at the end of last year, in which 3 different research groups had shown that they could actually use this system for correcting the mutation that causes a muscular dystrophy-like disease in a mouse model of this disease. So giving a lot of people sort of the idea that we're just showing the way, I think, towards the clinical application of this kind of technology. Still years away, for sure, but I think it's exciting to see that we're already to the point where this can be employed in animal systems for testing. But what about a different kind of application? What about an application where we actually make changes to DNA, not in fully differentiated cells or adult cells, but cells that are actually developing, a developing organism? We call this germline editing. And this is showing an example of germline editing that was done by Russell Vance, a colleague of mine at UC Berkeley, who was doing this in a fertilized mouse egg. And this is a pipette that's holding the fertilized egg over here, and we've got a needle coming in and injecting the editing molecules right into the cell. And then the DNA gets edited. And because this is a developing embryo, all of the resulting cells in this organism end up having a modification. And not only that, those changes can be transmitted to the next generation. Here's an example. Here was the outcome of this actual experiment is shown right here, where in this experiment, the Vance lab targeted a gene important for the black coat color in these mice, and the resulting pups that were born from that editing experiment, most of them actually had a double knockout. Both copies of that gene had been disrupted by the CRISPR-Cas9 system to create albino mice, and when the DNA from these mice was sequenced, they had to change at exactly the place that was desired, that was cleaved by Cas9. And they can transmit now that genetic change to their progeny. So, when this was an experiment Russell did in early 2013, and I showed this picture to members of my lab, who were people like me who are biochemists and structural biologists, and half of them said, "Wow, that's really cool." And half of them said, "Wow, that's really scary." And it was kind of both, right? Because you see very visually the power of this technology, but you also realize the profound nature of what's being done. And this really came home to me even more about a year later in early 2014, when I got a call one day in my office at Berkeley from a reporter who said that a scientific paper was about to be published in which a group had been able to do that kind of germline editing in monkeys, and had been able to efficiently change the DNA in monkeys in such a way that those changes can be transmitted. They were germline modifications. And it was really that experiment at that point that really motivated me to ask myself about how we as a society were going to grapple with this technology and how to employ it ethically and safely and appropriately in the future. And so I've been involved in a number of now global discussions around this question. What should we do now that we have a really exciting technology that allows precision genome engineering in effectively any kind of cell, including in germ cells and human embryos? And so about a year and a half ago now, with a number of colleagues, we held a meeting, a one-day meeting about this with a fairly small group of scientists from around the US, and published what we called a prudent path for employment of this genome editing technology in germline cells. And this piece really argued that scientists should voluntarily agree not to use the CRISPR-Cas or any other genome engineering technology in human embryos for clinical use, meaning to make a modified person, until the technology and and all of the ethical implications that go along with that kind of use could be fully discussed and considered. And so I've been really involved in this. I was very pleased that the national academies of several countries got together and organized a summit here in Washington in December of this past year to discuss this issue. And it's very much an active area of consideration right now around the world. In fact, a number of foreign governments have been involved in discussing this, and many, many different societies that have considered this issue and are thinking about how to proceed. And as we talk tonight, there are now 4 countries that I'm aware of that have actually approved the use of CRISPR-Cas9 for human embryo editing for research purposes. And those are Sweden, the UK, China, and Japan. So it's coming. And I think it's something that we all have to be really thinking about and understanding and considering what we think the appropriate response is in our society to an exciting and powerful technology. And I'll close by just acknowledging a great group of people in my lab. So this is a picture that shows my students at our 20th anniversary of my lab that was held 2 summers ago in my hometown, Hawaii. And which I was so happy, I guess. And then, we've had great collaborations, and I just want to end by pointing out that I think science today especially, really it relies on great collaborations because many of the projects we work on involve expertise that could never be contained in a laboratory, and that's certainly true in my case. And also, we couldn't do anything without funding, and I'm incredibly grateful to both public and private funding for giving us not a huge amount of money, but enough that we could get started on what we thought was a really interesting project, but had no idea of where it would lead. So thanks very much, and I look forward to the conversation we're going to have.
I've been told that we have roughly 700 people in the audience, so we can't all ask questions. However, I would like to invite people that are in the overflow room, if they have questions, Frank, I guess this is okay. Yeah, all right. They can't fire me. So I would say if there are people in the overflow room and you have questions, please come into the audience to ask, or to the main room to ask questions. So let's go ahead and start it. Do people have questions? If you do, please.
We talk first.
Got it.
That's why I got here so early. First of all, congratulations, both on all the work that you've done and on being a rock star, because now we know what you do in your spare time. This is fascinating. It's fascinating what you've discovered, and it's fascinating what you've developed, and it's fascinating how you tee up such a difficult question. Let's just start for a few minutes, though, on where you think this is going. You said in a few years, you pointed out Sweden, UK, China, Japan are already working on this. How far are we from clinical applications for human beings, do you think?
I think one point to make that I want to make sure people here really understand is that I think when we talk about clinical applications today, at least I'm talking about use in adults or kids, patients where we're treating a tissue. We're not creating changes that would be transmissible to future generations, not germline editing. There, I think the opportunities are truly exciting. I gave you the example of muscular dystrophy, but what I actually think is a more likely early target for this as a therapeutic is probably something like sickle cell trait, which is a disease of the blood, where editing can in principle be carried out outside of the body with blood that's removed from a patient, edited, validated that the cells are edited correctly, and then reintroduced. So I think that's where we're likely to see perhaps the initial
timeline on that. What do you think?
Well, there are a number of academic labs and companies that are moving in that direction, and I hear from my colleagues that people are talking about 12 to 18 months to file the first investigational new drug or IND request with the FDA. So we'll see if that happens, but if it does, then that means that they're getting approval to begin human clinical trials.
You talked about the work on monkeys, for example. What's that showing us?
I think it shows us that this technology operates in those cells as well as it does in other kinds of cells. And so that made me think, well, if you can do it in a monkey, you can probably do it in a human, and should we go there?
And?
Well, that's the question, isn't it? I mean, I think that, one of the things that emerged from the summit in Washington just a few months ago is that I think many people feel that, at least today, we mostly don't have the scientific knowledge to make changes to the human genome that would affect the kinds of modifications to a person that we might want. And so I think many people came away from that meeting feeling like, well, it may be our own ignorance of the human genome for now that prevents us from employing this technology in that kind of setting. But with the research uses that I mentioned at the end of the talk, I feel that it'll come. And so I think it's not too early for all of us to be understanding that this is a potential application, and is that a place that we want to go?
You talked about that crowd in your lab. Half of them said, wow, this is really cool, and half of them said, this is really scary. Which half were you on?
Maybe I sort of had one foot on both sides. Because I felt the same, you know, that picture literally showed up in my inbox, and the email was very short from my colleague, just saying, saying something like, wow, I can't believe this, or something like that, and I opened up the picture, and when I read about what he had done, I just thought first I thought, wow, that's just amazing, and then it really kind of seemed a bit awesome in every sense of that word.
I did a fair bit of research on you in addition to the conversation we had before I came over here, and I was watching one of your TED Talks. And in this TED Talk, you called for a global pause. You didn't talk about a global pause this evening. Still think there should be one? And what are we pausing to do? What are we stopping while we do it?
Yeah, well, I think what I mean by a global pause is a request for scientists to voluntarily decide that they will not use this technology for human germline editing, meaning human embryo editing, to lead to transmissible changes in humans. And I think that's very consistent with the consensus statement released at the end of the summit meeting in December. And the challenge is that that's really not something that can be, I think, enforced. It has to be agreed to by the scientific community.
So where is the scientific community on this now?
It's hard to say. I mean, science is a global effort now, and the thing to appreciate about this technology is that it's easy to obtain. It's available from a nonprofit called Agene, and people can trivially get these reagents if they want to do experiments. There's a company called Indiegogo that sells a kit to do CRISPR editing for kids. If you want to have your kids doing this.
This could be done by high school kids, right?
High school, or this is actually a kit for elementary school.
Elementary school. Imagine what they do to their
I don't know how
parents.
they sold. So it's easy to use, and so it means that we really can't, even if we wanted to, we can't put this genie back in the bottle. So I think we really want to think about how to go forward with it, but in ways that are safe and appropriate, and of course ethical.
No, it's really interesting because on the one hand, you have helped equip us, perhaps, to fight cystic fibrosis and cure disease, but some of the other things, what if this were used by bioterrorists? What if this were used to destroy food crops, right? So how do we navigate this? And is there a precedent for navigating this in science?
I think there is, actually. I think the precedent that I think is closest to what we're talking about here is actually what was called the Silomar meeting in 1975. That was a meeting that was to discuss the use of molecular cloning at that time. And people worried about applications of molecular cloning, making copies of DNA and doing that in bacteria. Would that be dangerous for humans and things like that? And I think, frankly, with any new technology, there's always the risk of inappropriate or dangerous applications. And CRISPR is no different. But I do think that it's very important to be grappling with these issues now. And a little anecdote that I think about in this context is when I was, at my sort of in the early months of doing this work, realizing that sort of this joy of discovery, really, of figuring this out and understanding how this worked and thinking it was really interesting and really cool, and then seeing the pace of science just increasing, tremendously. It's hard to overemphasize it. I mean, now there's close to 2,000 publications in the scientific literature in the last 4 years that are using this technology. So the pace just took off. And I had this incredible, disconnect between my professional life, going to work and reading papers and talking to my colleagues and my students and all the people that were using this for various things. And then going to PTA meetings, going to school functions with my son and his friends and their parents. Most of them were not scientists, and realizing they had no idea about this, and realizing that there was this important technology that people really needed to start to understand so they could think about how it might be used in the future.
What I'm going to do in about 5 or 6 minutes is open it up to some questions. There will be mics on either side, and I'll invite you down. But I want to, before I do that, come away from the ethical dimension for just a moment and come to this notion that we could be on the threshold of really revolutionizing medicine because of all of this. Close your eyes for a moment and imagine where this could take us. And presuming that we can navigate through some of this ethical swamp, what the world could look like in 10 or 20 years from now.
I think there's a few areas where I see, really exciting impacts that affect human health and human society. Human health, sort of broadly defined. I think that, John Holdren's, one of his slides pointed out that one of President Obama's real scientific initiatives has been the idea of personalized medicine. And I think that this technology is perhaps one of the ways that we can really get there, by understanding for each of us, what our genetic makeup is, what kinds of diseases we might be susceptible to, and where there might be opportunities to make changes that would be preventative, perhaps, and certainly to do things that will correct disease-causing mutations in the future. And we've already sort of discussed that. But I think it's also exciting for me to think about applications in agriculture. I think that opportunities to make changes in plants that will protect them from pathogens in the environment or from environmental climate change and that sort of thing. I think that's also very important, as well as opportunities in sort of the area I would call synthetic biology, very broadly defined, and being able to change the DNA in industrially important organisms, like industrially important yeast, or introducing chemicals that are produced in a sustainable fashion. I think these are all areas where we're likely to see big advances.
And what about, for example, with driving genes for malaria-carrying mosquitoes that could render them extinct and the disease as well?
Yeah, that's right. Something called gene drives that George Church and others have been, really, I think, rightfully pointing out is an exciting but also equally sort of a little bit scary kind of application where one could use a very efficient genome editing technology like this to drive a trait through a population very quickly.
Through an entire species.
Through an entire species. And people are already thinking about using this to control mosquitoes. In Brazil, for example, I know the Gates Foundation has been very interested in that, and other groups as well. So I think, again, that's a question of, is that wise to do? And if we do it, how do we make sure we can control it?
Sure, the question raises, what is the environmental impact if you make an entire species go away, or if you drive that gene through an entire species? I was going through some of the clippings and things, and Madeline Jacobs sent me a lot of these gene editing. It's all over the media, right? Which is what I do, so I'm kind of curious about this. Gene editing offers hope. I like to say I'm from the media and I'm here to help. Nobody believes that. Gene editing offers hope for a severe form of muscular dystrophy. Here's something, editing the genome. Making the cut, CRISPR genome editing technology shows its power. It talks about here how high school kids can get it, which I find truly thoroughly terrifying. Gene editing, great potential and great risks. And in the great potential and great risks, it talked about these gene drives and talked about some other things. How do you think we have a grown-up conversation at a time when we're not very good at having grown-up conversations in this country about something that is so profound and profoundly difficult?
I turn to the media. Well, I think, no, I'm joking, but I'm not. I think the media...
You come out of the lab to take a very serious position on it.
I think it's important that scientists do that, and I have come to that only with some reluctance, I have to say, because, as scientists, I think we're naturally, we want to do the next experiment in the lab, and that's the thing that we sort of, gets us out of bed in the morning. But I do think that that when I think about the way that science is seen in our society, and the story that we were talking about on the way to this lecture was the example in the news recently of someone who saw a scientist on a plane doing working equations and thought this person was a terrorist and had been pulled off the plane. I mean, really,
You got some work to do.
sort of, yeah, we have work to do. And, and I think just if you think about the reactions of the public to certain things like climate change, to vaccinations, to genetically modified food, I think these are all areas where, frankly, there's been a lack of a real disconnect between what scientists are doing and what people who are non-scientists understand.
What's the reaction you got? What's at the PTA meeting? How do people respond to you and what you've done?
They're amazed. I think they're just, right now the response I've gotten is mostly just kind of amazement. They're interested. They're struggling to understand it. They're trying to really grasp what this really means. I take my 3D printed model is actually a sort of a football-sized thing, and I found that to be a very useful, sort of prop in a way to explain this, to help show people what this technology is really based on. But I think that, many people, and I have to say that I get contacted, and I'm sure other people working in this field do the same thing, but I get contacted every day, every single day, by people, parents, patients, people that have genetic disease in their family, and they are desperate. They want to understand, when will this be employed to help me, my kids, my family? And it really touches me profoundly because, and I have to explain to them, we're still clearly years away from using this therapeutically, but nonetheless, just for many of them, just seeing that this is coming and may come in their lifetime is very, very exciting.
The therapeutic uses obviously are very exciting. The enhancements are another question. And you've talked about designer humans that could come out of this. Real possibility? Something you worry about?
Well, again, I guess I go back to, on the one hand, yes, in the sense that this technology is likely to enable those kinds of changes to be made in embryos were we to want to use it that way. But I do think that at least where we are right now, we don't understand, for the most part, enough about the human genome and the interactions between genes to know how to make changes that would have enhancements that might be desirable in the vast majority of cases. But I do think that will change over time, and especially now that research is beginning using this in embryos in some jurisdictions, that will will drive forward our understanding, and that will, I think, certainly motivate people to want to use it clinically in some settings.
Let me invite you to move to the microphone, and I'm going to turn to some of your questions. And this does not look like a shy audience. And so you're there first. Why don't you identify yourself? And if we can keep the questions fairly short, we can get as many in as we can in the remaining time. Go ahead.
Joanne Oravec from the University of Wisconsin. I've got intellectual property related questions about how corporations, other entities might profit from this research. And given the huge investment of our nation, the National Science Foundation, other agencies in this gambit, is that fair? Maybe we as a nation should somehow benefit and not allow corporate entities, whatever, profit-making organizations to exude profit. And well, if changes are made to my genome, will I have to pay over time, like a subscription, in order to keep living? So thank you so much for your research.
Wow, where to start with that one?
Let's start with corporations and profit.
I guess the short answer to that question is that right now, for academic researchers like me, the intellectual property that's created during research efforts is owned by our universities. So it's the universities that, basically are responsible for filing patents and then defending those patents and licensing those patents. And as you probably appreciate, having patent protection has long been appreciated as being very important for commercialization. And am I in my own lab going to be able to develop a therapeutic from this? The answer is no. We don't have the resources or the wherewithal to do that. It really has to be done in the context of a commercial entity, and they won't be interested in doing it unless they know they can ultimately make a that's sort of our standard patent system. But I think you're right to bring up this point about, it's sort of a rhetorical question, but one we're thinking about. How should publicly funded science be, how should profits from that be fed back to the public?
Go ahead, sir.
Given the potential for abuse of this technology, do you see any way that there might be a countermeasure? Is there a way to interfere with the action of CRISPR to recognize, et cetera, in some way?
The short answer is yes. So, and many groups are working on exactly that, how to regulate this function of this protein, how to shut it down, how to reverse its activities. Those are all, I would say, it's very much sort of at the bleeding edge of the research right now, but I think there's a real appreciation that would be a useful thing to be able to do.
Sir.
My name is Martin Apple, and I'm the past president of this Council of Scientific Society Presidents. I would think the logical approach for us to take here is to say that we can do certain medical goods in humans, and that should be number one priority. And we've exhausted that possibility. Give us how many years it takes to do that. We can then have developed the time to develop an adequate standard of ethics and to make that standard of ethics worldwide. Rather than speculate on it now, take an elbow here and a finger there and so forth, and everybody do something different. This is one time in human history in which we really have to have a universal standard of ethics agreed to, and we haven't gotten there. But we can do a lot of good on the way, and that can set the groundwork for doing it.
What do you think?
Yeah, is that a question or?
Observation, you have a response.
Yeah, an observation. I mean, so I certainly agree with you. I think the challenge that I see is how do you enforce that globally? If there are people in some jurisdictions that don't necessarily share that view, and what do we do about labs that are working in those parts of the world where they can easily get this technology and they can do human embryo editing if they want to?
And where systems of transparency and accountability don't
Right.
come anywhere near what we're talking about here. So
Right.
it's a very difficult
Right.
question.
I see that as a challenge.
Yes, ma'am.
I think I understand. I don't have any scientific background in this area, so I think I understood what you were explaining about the cutting part of the. There's two phases, and then the second phase was somehow either the two loose ends connected to each other, or they connected via some kind of a designed bridge sequence, and either you didn't really explain that or I somehow missed all that. So I don't know how you get that little bridge sequence in there after you've done the clipping.
Yeah, no, it's actually a great question, and I didn't explain where that comes from, but it really can be introduced in a couple of ways. One is by the experimenter putting that piece of DNA into the cell so that it can be incorporated during the process of repair. Or sometimes cells, and this is what they do naturally, is if there is a piece of DNA in the cell that has a sequence of letters that matches the site that got cut, they can actually use that as the source of insertion of a new sequence. And that actually is a process that often happens naturally in cells, like during DNA replication or cell division. And so it can come from inside the cell or externally.
Sir.
Thank you for a fascinating presentation. I've got a couple of technical questions. One is that you mentioned for treatment of sickle cell syndrome, you could remove an aliquot of blood, treat it with CRISPR, give it back to the patient, but then the blood cells overturn. Okay, so that'd be a sort of a temporary fix. Related to that, do you see that there will be a possibility to do CRISPR editing at the gamete stage?
Okay, so your first question was, how does this work if we edit blood cells they're going to turn over? And you're right, and so the way that this would be used therapeutically is by editing blood stem cells, right? So these would be cells that have proliferative properties. They can repopulate over time the blood system. And so it would really be at the level of stem cell editing where this would be, I think, most valuable therapeutically. And your other question was, gametes. Can you use it in gametes? Yes, you can, or at least, we think so. And I think that's actually a fascinating idea. There was a wonderful presentation at the summit in this past December from someone at Pittsburgh working on sperm and the opportunities there to make therapeutic changes in sperm. I think it's a really, again, there's lots of research to be done, but it seems like an exciting opportunity.
Yes, ma'am.
Hi, Flora Larenman, and I was wondering, what are the predictions that the scientific community might have for how the edited genes will withstand the longitudinal effects of epigenetics?
Well, I don't know quite how to answer that. I mean, I think that if you're talking about, how will they respond to? Are you talking about methylation that might turn them off, or
Yeah.
well, it's interesting that so in a just a sort of anecdotal kind of answer to that question. Experiments that I've done with a colleague at Berkeley, Dirk Hockmeyer, in human stem cells, what we're seeing is that when you make changes to those cells, there are epigenetic modifications that occur sometimes to those inserted genes that turn them off. And so I think certainly that's obviously going to have to be an active area of research to understand what happens to genomes that get edited and how is the new information in those genomes controlled by epigenetic modification. I don't really think we know the answer yet. Well, I don't know the answer.
Sir.
What I guess, innovations do you hope to see in the next generation of research into this? And I guess second question would be, what new types of businesses do you, I guess, expect or hope to see evolve from this kind of like 23andMe evolved from the mapping of whatnot?
Yeah, well, I think that sort of very broadly speaking, I think we'll see a couple of kinds of businesses. One is sort of technology companies that will build on this and new things that come along and sort of that are in this kind of super family of proteins that do make DNA modifications, and companies that will sort of develop those as platform technologies. And then I think we'll see companies that are actually specializing in applications, whether it's in agriculture, whether it's in therapeutics. There's a new company that's involved in animal modification to make animal organs that are better for organ donation. I think there will be lots of creative things that people come up with to use this to do. There's even a company I heard about recently that is trying to make a hypoallergenic cat. So stay tuned to that one.
The naming opportunities there are really quite fun. I'm told we have time for just one more question, so sir, that's yours.
Thank you. I'm going to try to draw an analogy between this and years ago when there was this whole issue about embryonic stem cells and what could be used for research and what couldn't. And that was tied up in a very volatile issue, which was abortion. And this is not. But the government chose, if I'm choosing the right word, to regulate that. And there were permissible things you could use, and there were non-permissible things. Is anyone moving in that direction? Although this is a very different area that doesn't involve all those issues, it still has lots of ethics. Is anyone moving in the direction of trying to regulate this from a government point of view?
Well, that's probably more of a question for John Holman than for me. But again, I guess what I come back to is that it's hard to regulate, I think, right now. I mean, certainly you may know that federal funding cannot be used right now for human embryo modification in this country, right? But that being said, if a private foundation or state money were to be employed for that, that would not be illegal in this country. And so, how to grapple with the challenges that may come up going forward, given that sort of situation, I think it's a fascinating question. I've actually been already a couple times to the Office of Science and Technology Policy to discuss that issue, and I think it really has to be an ongoing conversation.
Jennifer, before I hand this over to Dave and we pack it in for the night, I want to ask you to sort of reflect on a personal scientific level for all of us. We hear so much about the joy of discovery, the amazing moment. Would you take us back to when this was all getting real for you, and what you thought about and what you reflected upon, if you did, that you
Well, it's like, I guess when I reflect back on it, I feel like I just reflect on the very humble origins of this. That this was really as you maybe saw from the way I presented this, a curiosity-driven project. It was being worked on by just a few people in my laboratory at the time, and a few people in labs elsewhere. So it was a really small field of people, and I think for me, just getting to the point where we understood that bacteria have evolved an incredible machine for, recognizing viral DNA and destroying it. First of all, that was the pure joy of discovering. Wow, that's so amazing that nature does that. And it's so cool. I just thought that was really an interesting thing about bacteria. But then, for me, making that connection to how it could be harnessed as a technology was sort of just a moment of taking understanding and realizing that it could be connected to something that you never imagined it would be connected to. And that's something that I've always liked about science. I think that's really fun, is that, you could be working over here and suddenly realize that your results or your insights have to do with something over here. And, and I think this is really an example where that was the case. And, it was just that sort of proverbial moment of, chatting in my office with Martin Jinek, the guy that was actually doing this work in my lab, and realizing that, this could be a really interesting technology. And the two of us, literally kind of looking at each other and, feeling the hairs on the back of my neck, because it really seemed like an exciting moment. So.
Well, I hope you share that with your students and the others who come through your lab. So thank you so very much.
The sense of wonder. That's the second time it came up today, which I think is wonderful. Thank you so much, Dr. Doudna. Thank you, the Carnegie Institution, for hosting this magnificent place. And thank you all for coming tonight. Good night.